For this video experiment, we will be using 3 sample proteins: casein, albumin, and gelatin. Samples for each of the solution will be placed in 3 separate test tubes. The first qualitative test is the Biuret test. This qualitative test is a general test to detect the presence of peptide bonds which connects amino acids to build proteins. And the procedures are as follows. For this qualitative test, it starts with the addition of 1 ml 10% sodium hydroxide solution to each of the protein samples placed in 3 separate test tubes. After which, 1 to 3 drops of 0.01 molar copper sulfate solution is added to each of the mixture in the 3 separate test tubes. Observe the color produced after the addition of 0.01 molar copper sulfate solution. The second qualitative test is Ninhydrin test. This qualitative test is used to detect alpha amino group of proteins or free amino acids giving a blue or purple colored complex. And the procedures are as follows. First, we add 5 drops of fresh Ninhydrin solution to each of the sample proteins. After which, we put these test tubes in boiling water bath for 10 minutes and observe the results. The third qualitative test is Xanthoproteic test. This qualitative test is used to detect the presence of amino acids containing an aromatic nucleus in the protein solution, which gives a yellow-colored nitro derivatives on heating with concentrated nitric acid. And here are the procedures. In this test, 1 ml of concentrated nitric acid is added to the protein samples. Then we place this test tube in boiling water bath for 30 seconds and let it cool down in a rack after. After it cooled down, we add drop by drop saturated sodium hydroxide solution to each until the solution becomes alkaline. And here are the results. The next qualitative test is Millon's test. This qualitative test is used to test tyrosine or proteins containing tyrosine that will give a red color or precipitate when present. And here are the procedures. Here, we add 5 drops of Millon's reagents to each of the sample solution. Then we heat the mixture in boiling water bath for 5 minutes. Remove the test tube from the water bath and let it cool. And here are the results. The next test is Sakaguchi test. This qualitative test is used to test the presence of arginine or arginyl residue giving a red colored complex. And here are the procedures. In this test, we add 1 ml of sodium hydroxide solution and 1 ml of 0.02% alpha-naphtol solution to each of the samples. After 3 minutes, we add 2 to 4 ml of bromine solution. Observe the color produced. And here are the results. The next qualitative test is Hopkins-Cole test. This test is for the presence of tryptophan. A violet color between the 2 layers indicates a positive test. Here are the procedures. Here we will be adding 2 ml of Hopkins-Cole reagent to each of the sample solutions. After which, we add 1 ml of concentrated sulfuric acid. Let it stand for 1 to 2 minutes and observe. Take note of the color produced between the layers. And here are the results. The last test is sulfur reaction or lead acetate test. This test is for the presence of cysteine or any proteins containing these amino acids. A black or gray precipitate indicates a positive test. And here are the procedures. For this test, about 5 ml of 5% sodium hydroxide solution is added to each of the sample. After which, few crystals of lead acetate is added to each test samples. We heat the mixture in boiling water bath for 5 to 10 minutes. Then remove it from the boiling water bath and let it cool down. After it cooled down, These are the results.